Hello everyone, welcome to our YouTube platform Concept and Fact of Life Sciences. Today in this video, we are going to discuss about the oxidation of fatty acids. Oxidation of fatty acid, it is mainly are of three types: alpha, beta and omega oxidation. It is simply the structure of a fatty acid. It is having more than two number of carbon, so it is simply known as the acyl chain. The carbon which remain present next to the functional group, it is called as the alpha carbon followed by a beta carbon, and the carbon which remain present on the other terminal, it is known as omega carbon. The oxidation, it occurs on the alpha, beta and omega carbon, known as alpha, beta and omega oxidation respectively. First we take the alpha oxidation. This alpha oxidation, it generally occurs within the peroxisomes. It is mainly involved in the oxidation of a phytanic acid. Phytanic acid, it is a 20 carbon fatty acid. In first step, there is addition of hydroxyl group onto the phytanic acid, lead to the formation of alpha hydroxyphytanic acid. Then there is a removal of a methyl group from there, that lead to the formation of a pristanic acid. So there is a conversion or oxidation of phytanic acid into the pristanic acid by the removal of a terminal methyl group, by the removal of a terminal methyl group. If there is a absence of this conversion, that is the conversion of phytanic onto pristanic, or we can say there is an absence of this alpha oxidation, then there is an accumulation of this phytanic acid within the peroxisome that lead to a disease condition known as Rapsum disease. So this is the alpha oxidation. Next is the omega oxidation. We are taking omega over here because the major type of oxidation in case of both plants and animals, it is the beta oxidation, जिसके बारे में अपन अच्छे से बात करेंगे. So first we are taking the omega oxidation. During the oxidation, it occurs, beta oxidation, it occurs within the peroxisome and the mitochondria. But this type of oxidation, that is called as the omega oxidation, it occurs within the endoplasmic reticulum. The oxidizing environment which is provided by the endoplasmic reticulum, it is a omega oxidation, so it will occur onto the omega carbon. So this omega oxidation, it oxidizes the terminal omega carbon. As we can see over here, it is the omega carbon. For this oxidation, the endoplasmic reticulum, they are having an enzyme which is known as cytochrome P450 monooxygenase. There is a cytochrome P450 monooxygenase enzyme which adds the oxygen atom onto this acyl chain. Simply, it is acyl chain, as we already stated. It is the omega carbon. On this omega carbon, there is a functioning enzyme which remain present in the endoplasmic reticulum, that is cytochrome P450 monooxygenase. It adds the oxygen atom onto this omega carbon, leading to the formation of omega hydroxy acyl chain. So there is an addition of this hydroxyl group to the acyl chain. Now this cytochrome P450, it is mainly involved in the detoxification reaction that occurs in the endoplasmic reticulum. So it metabolizes the xenobiotics. This omega oxidation, it metabolizes the xenobiotics. Xenobiotics are generally the toxic compounds. So here there is an oxidation of xenobiotics. In first step, there is an addition of oxygen atom and conversion of this acyl chain into the omega hydroxy acyl chain. Then there is a functioning of a dehydrogenase enzyme which further oxidizes this omega terminal, leading to the addition of a carboxylic group onto the omega terminus. So now this fatty acid, it is having carboxylic terminus on both of their ends that can be sent to the mitochondria for further oxidation. Now the oxidation of this xenobiotic or the large chain fatty acid, it occurs from both ends. So there is a neutralization of these fatty acid, and the initial process, it occurs within the endoplasmic reticulum with the help of enzyme that is called as cytochrome P450 monooxygenase. So this is all about the omega oxidation that is involved in the we can say detoxification or neutralization of the xenobiotics. Now beta oxidation, it occurs in case of plant and animals both. It occurs both in case of plant and animals. In case of the plant, the beta oxidation, it occurs within the peroxisomes. The plant, they are having a special type of peroxisome that is called as glyoxysome. The product of beta oxidation in case of both plants and animal, it is the acetyl coenzyme. Now, in case of plant, this acetyl coenzyme A, it can directly participate into a cycle which occurs within the special type of peroxisome that is known as glyoxysome, and the cycle is known as glyoxylate cycle. Now, it is a modified version of TCA cycle that converts this acetyl coenzyme A directly into the sugars. So, the plant, they have ability to generate sugars directly from the fatty acid. In case of animal, the beta oxidation, it occurs within both mitochondria and peroxisome. Major जो beta oxidation होता है, that occurs within the mitochondria. Initial trimming, it occurs within the peroxisomes. The product, again, it is the acetyl coenzyme A. But in case of animals, there is no glyoxylate cycle, or there is no glyoxysome. So this acetyl coenzyme A, within the mitochondria, we are having a cycle in the mitochondrial matrix that is called as the TCA cycle or the Krebs cycle. This Krebs cycle, it converts this acetyl coenzyme A, or we can say the acetyl coenzyme A, it can participate in this TCA cycle, leading to the formation of energy in the form of NADH and FADH. These NADH and FADH, they gives their electron to the electron transportation chain, and there is a generation of energy in the form of ATP. That is the energy currency. If we talk about the procedure of beta oxidation, now first step in the beta oxidation is the activation of a fatty acid. It is simply the structure of a acyl chain or the fatty acid having more than 2 number carbon, or we can say n number of carbon. It is known as acyl chain. The first reaction, that is the activation, it is carried out by an enzyme which is known as thiokinase. Now, whenever there is an involvement of thiokinase, there is a utilization or the production of ATP. Here, the ATP will convert into the AMP, and there is a release of pyrophosphate that lead to the conversion of this acyl chain into the acyl coenzyme A, because at the same time we are also adding a coenzyme A. So this acyl chain will convert into the acyl coenzyme A. We simply added this coenzyme A over here with the help of enzyme thiokinase. ATP here, it is converting into the AMP, so it is utilizing 2 ATP equivalent energy. काम आएगा जब अपन एनर्जी बजट बनाएंगे इसका. So we are having acyl coenzyme A. This is the activation reaction. Now after activation, we have to transport this acyl chain into the mitochondrial matrix, and mitochondria not allow the transportation of acyl chain in the form of acyl coenzyme A, so we have to convert them into another form. Now for this, we are having an enzyme which is known as carnitine palmitoyl transferase. Because in the majority of time, there is a transportation of 16 carbon palmitic acid. So we are having CPT, carnitine palmitoyl transferase 1. We are also having its isoform in the mitochondrial matrix, that is called as CPT2. Now this CPT, it removes the coenzyme A and will add the carnitine onto this acyl coenzyme A. There is a removal of coenzyme A, addition of carnitine, lead to the formation of a acyl carnitine. Now this acyl carnitine, it can be transported into the mitochondrial matrix. So we are having the second step, that is the transportation to the mitochondrial matrix. It will be carried out by the enzyme CPT1 and CPT2. CPT1 convert the acyl coenzyme A into the acyl carnitine. It can be transported to the mitochondrial matrix. Within mitochondrial matrix, we are having the carnitine palmitoyl transferase 2, which removes the carnitine, will add the coenzyme A. Now again we are having the acyl coenzyme A. So जो ये कार्निटाइन कैरियर का यूज किया गया, ये जस्ट आपके एसाइल कोएंजाइम ए को माइटोकॉन्ड्रियल मैट्रिक्स में ट्रांसपोर्ट करने के लिए किया गया. So the second step is the transportation of acyl chain into the mitochondrial matrix. Next is within the reaction which occurs within the mitochondrial matrix. Now there are four important reactions which are oxidation, hydration, oxidation and thiolysis. First we talk about the oxidation. We are having acyl coenzyme A within the mitochondrial matrix, and we are having an enzyme which is known as acyl coenzyme A dehydrogenase. This dehydrogenase, it is FAD specific. It removes two hydrogen from this acyl coenzyme A, one from the alpha carbon and other from the beta carbon. So there is a formation of a double bond over here because the carbon valency is 4. So we are having 4 valencies. Now this structure is known as the enoyl coenzyme A. In the second step, first step was the oxidation. We oxidizes the alpha and the beta carbon both, leading to the production of a enoyl coenzyme A. Second step is the hydration. We add the water molecule onto this enoyl coenzyme A. Now we can write the water molecule in this form also. So we will add the OH group onto the beta carbon and H group onto the alpha carbon in this way. Now this structure, it is called as a beta hydroxy acyl coenzyme. We are having a hydroxyl group onto the beta carbon, so it is a beta hydroxy acyl coenzyme A. Then there is a functioning of enzyme which is known as beta hydroxy acyl coenzyme A dehydrogenase. Again there is an oxidation reaction. Now it is NAD specific, so convert the NAD into the NADH. Now this time both of these hydrogen, they will be taken up from the beta carbon because we are studying the beta oxidation. So there is a removal of 2H from the beta carbon, sorry. So that will lead to the formation of a structure which is called as beta keto. Now there is a removal of 2H leading to the formation of a keto group. So beta keto acyl coenzyme we are having. The fourth reaction is the thiolysis. For this we have an enzyme which is known as thiolase. It will add a coenzyme A to the beta keto acyl coenzyme A, leading to the formation of acyl coenzyme A having 2 less number of carbons. So we just simply added this coenzyme A over here that lead to the formation of acyl coenzyme A having 2 less number of carbon, because we are also having here a acetyl coenzyme A which is of 2 carbon. अब जो ये 2 कार्बन का acetyl coenzyme A बना है, it will enter, or we can say it will participate in TCA cycle within the mitochondrial matrix, that is also known as the Krebs cycle. Now through the Krebs cycle, from 1 acetyl coenzyme A, we will get 3 NADH, 1 FADH and 1 ATP. And when this NADH gives electron to the electron transportation chain, which also occurs within the inner mitochondrial membrane, then from 1 NADH we are getting 3 ATPs. So from 3 NADH we are getting 9 ATP. From 1 FADH we are getting 2 ATPs. So there is a production of 2 ATP. 9 plus 2, 11 plus 1, 12. So from 1 acetyl coenzyme A in the TCA cycle, we are getting 12 ATP equivalent energy. समझने की कोशिश करते हैं. If there is an oxidation of a palmitic acid, as we already stated, it is a 16 carbon fatty acid. If it enter into the beta oxidation के कितने rounds चलेंगे? There is a 7 cycles of beta oxidation, because 7 cycles, in 7 cycle you will get 2 acetyl coenzyme A molecule. So there is a 7 cycle of beta oxidation. Due to the 7 cycle, you are getting 7 NADH and 7 FADH. From 1 NADH, you are getting 3 ATP. So from 7, you are getting 21. From 1, you are getting FADH, you are getting 2 ATP. So from 7, you are getting 14. So the total number of ATP, it will be 35. Now, as a product of beta oxidation, you are also getting this acetyl coenzyme A that can participate into the TCA cycle, and in TCA cycle from one acetyl coenzyme A you are getting 12 ATP. So from 8 acetyl coenzyme A you will get 96 ATPs. So 35 plus 96, it will be 131. Now, during the activation, we already stated that there is a utilization of 2 ATP equivalent energy here. So, there is a utilization of 2 ATP equivalent energy. So, we will deduct these 2 ATP from 131. So, net gain from the palmitic acid oxidation, it is the 129 ATPs. So, इस तरीके से आप किसी भी फैटी एसिड के ऑक्सीडेशन से आपको कितनी एनर्जी एटीपी के फॉर्म में प्राप्त होगी वो निकाल सकते हैं. Another important enzyme of beta oxidation, it is the carnitine acetyl transferase. It is not acyl, it is a acetyl transferase. Now, within the mitochondrial matrix, if the glucose level are high, then you need not to generate energy from the fatty acid. You are having glycolysis, Krebs cycle, and the plenty of glucose, so that can participate in this oxidation reaction and produce huge amount of energy. So you are in the energy rich state. During this state, the acetyl coenzyme A, which produces upon the beta oxidation of fatty acid, it will accumulate within the mitochondrial matrix because you are having plenty of glucose in the matrix. So during these conditions, there is an enzyme which is called as CAT, carnitine acetyl transferase. This carnitine acetyl transferase converts this acetyl coenzyme A into the acetyl carnitine. By removing the coenzyme A and adding the carnitine, there is a production of acetyl carnitine. This acetyl carnitine can be transported into the cytoplasm. So we can say it do not allows the accumulation of acetyl coenzyme A within the mitochondrial matrix. So this is all about the beta oxidation process. Thank you.